Welcome to the Minnesota Stormwater Seminar Series. My name is Andy Erickson. I'm honored to host the seminar series today. I'll be introducing our keynote speaker here in a moment. But first, I want to thank those that participate and help make this seminar series possible. Minnesota Stormwater Seminar Series is funded by the University of Minnesota Water Resources Center in partnership with the Minnesota Stormwater Research Council, Clean Water Land and Legacy Amendment. We partner with the Minnesota Pollution Control Agency, and we host right here at St. Anthony Falls Laboratory as part of the University of Minnesota. We're super excited to have you here with us today. We are going to have a great seminar and panel discussion. The topic today is addressing the challenge of water-soluble emerging contaminants in green stormwater infrastructure. As I said, we'll start with a keynote presentation by our invited speakers, and then we'll follow up with a panel discussion, including local experts on our topic today. But without further ado, I'm going to introduce today's keynote speaker. Dr. Greg LeFevre is an associate professor of environmental engineering and science in the Department of Civil and Environmental Engineering and IIHR, Hydro Science and Engineering at the University of Iowa. Greg and I didn't really overlap. He went to Michigan Tech, started at Michigan Tech just as I was leaving, go Huskies. He, after finishing at Michigan Tech, came here to the University of Minnesota to do his graduate work, then did a postdoc at Stanford, and has been at the University of Iowa for 10 years, I think, right? Almost exactly, pretty close, all right? So Greg's been doing a lot of work in this area of stormwater and emerging contaminants, many publications. If you do not follow Greg on LinkedIn, I encourage you to do so. It's really exciting to see the stuff he puts out on LinkedIn. But today he's going to be talking about addressing the challenge of water-soluble emerging contaminants. So without further ado, please help me welcome Greg to the stage.
Well, thank you very much for the kind introduction, Andy, and I'm really excited to be here and honored to be part of this series. It's kind of, the epicenter of the stormwater world, and I'm really proud to be here as an alum of this program and everything like that too. So I'm excited to tell you about the research that our lab has done looking at addressing the challenge of water-soluble emerging contaminants in green stormwater infrastructure. And there we go. Here's a picture that Ray took like 10 years ago. It's really cool, right? And you probably saw some of these walking here just this morning. It's inescapable that land use practices that develop natural and agricultural areas into more impervious surfaces results in an increase in the runoff potential as well as the contaminants that are present in that stormwater. Sometimes it's really visible, like rainbow sheens through stormwater. Sometimes it's not visible. And the old paradigm used to be that if you remove the particles, you remove the pollutants. And that was what it was for decades. But now we've gotten a much greater appreciation in the last decade or two about what is also in those things. And so I'm proud to be part of a group here that probably this was officially published in, I think, 2015, and a lot of folks here are in this room, looking at dissolved pollutants and their importance in stormwater. And this includes things like dissolved nutrients, dissolved metals, and organic contaminants as well. So the USGS, the U.S. Geological Survey, has also been instrumental in going in and looking at stormwater as a complex mixture of organic contaminants and in stormwater and how they move and what is present in those systems. Okay. What do we mean by emerging contaminants? There's not really a good definition of what that means necessarily, but many of these things include things that are soluble, right? So they're dissolved in water, and so that means they can be transported very readily from their sources. And organic contaminants are things oftentimes like urban use biocides, like pesticides, including insecticides, herbicides, urban use biocides that are impregnated into the sides of buildings to prevent mildew and molds, vehicular-related compounds like tire wear compounds, anti-corrosives. So oftentimes these are present as complex mixtures, and we don't necessarily know how they work and interact all together. And for some things like pesticides, they're designed to kill things. So that has obvious biological impacts on the receiving water. None has really gotten more attention in the last few years than 6-BPD-quinone. This is actually an oxidation product of an antioxidant that's added into tires, and then when these wear down on the roadsides, then they can be washed into streams, and when they're oxidized, it can actually, at like sub-nanogram per liter levels, cause what's referred to as urban mortality stream syndrome for coho salmon and other salmonoids as well. So it's an acute problem in the Pacific Northwest. So again, go back to that remove the particles, remove the pollutants type of thing. What do we know about this, right? We know that bioretention cells and other green infrastructure is designed to infiltrate stormwater pretty rapidly. That's, we're trying to optimize for multiple different dimensions here. And so if we're looking just at the hydraulics, a conventional system tends to have pretty sandy media to increase the hydraulic conductivity, make it so that water flows through here pretty fast, right? We want to increase the infiltration. But we do get a lot of removal of particle-bound or particles, like sediments, as well as particle-associated contaminants, like more of those hydrophobic things that might be stuck onto particles. But new research is demonstrating that bioretention cells in conventional systems are not using conventional sandy media is not particularly good at soluble emerging contaminants, right? I know which makes sense. If it's traveling with the water and the water is being moved through the system quickly, it's not going to be removed very well. So this brings us to what I see as the central challenge of emerging contaminants in green stormwater infrastructure, is that a lot of these emerging trace organics are very soluble, right? And so they're going to be moving with the water, and we want to maintain this high hydraulic conductivity so that we can maximize infiltration. But if they are moved through, that could potentially impact the groundwater and beneficial reuse of that. If we go in and somehow are able to go in and just have them all stick into our cell, that could potentially mean that they could accumulate in there, which is not also good either. So really this brought us to a couple of important questions, which is what I'm going to be talking about here in my talk today, is can we sustain removal of contaminants using biological processes, right? Things like, bioremediation is using bacteria, plants, fungi to go in and actually degrade them and degrade them in situ, right? This is a self-sustaining, low-maintenance approach to going in and addressing these contaminants. Can we improve the geomedia to make it so that we're better capturing the contaminants during this rapid infiltration and then subsequently biodegrading these contaminants as well? And what types of novel measurement approaches can we use to characterize these complex mixtures, right? So that it's not just thinking about this chemical soup that's there and what is it, right? So we're not just taking a one-by-one approach. So here, the first part here, I'm going to be looking at biological processes in green stormwater infrastructure with the goal of sustaining contaminant removal. I'm not sure why it's keeping on going black here. So I'm going to harken back actually to, I did my PhD here, right? And this is something I've been working with stormwater and been excited about it since I was an undergrad. This is, deep in my roots here. And, classical bioremediation is working with bacteria. So some of the work that we did here was looking at how bacteria in bioretention cells can go in and limit the accumulation and make it so that we can go in and sustain removal for, in this case, petroleum hydrocarbons. So this is cool. This is really cool, right? We also looked at some effects of plants, right? So I'm actually not going to talk about bacteria, even though I think that they're very important. I'm going to talk about some of the work that we've done more recently that is looking at what I consider to be a little bit more of the neglected biological processes, because bioremediation is, we classically think of that as bacteria. I'm going to start out with vegetative processes, right? We know that we published a cool review, and you can download that and stuff like that, looking at vegetative processes in bioretention. We know that there are hydrologic benefits by going in and maintaining hydraulic conductivity associated with the roots and avoiding compaction, preferential flow paths. We know that there's water quality benefits by going in and being able to take up contaminants. That's kind of what I'll unpack a little bit more in detail here. We know that there's what we call generally ancillary benefits, especially aesthetics and improving the livability of neighborhoods. And then there's also, certain plant traits that make plants more or less desirable for inclusion in there. And these things include their root structure and stuff. But one of the questions that's often asked is, can phytoremediation, right, using plants to remediate this, can this be practical for a situation involving stormwater, right? Remember, we want to maintain fast infiltration, high hydraulic conductivity. Can this be relevant? So kind of the way that we think about phytoremediation and the way that that entire research field was developed was thinking about these legacy contaminants. And kind of the paradigm of that was that the contaminant would be taken up into a plant at the same rate at which the water was being taken up, right? Transpiration. That would be the theoretical maximum because the contaminant can't be taken up any more than the plant, or the plant is taking up water. And because there's membranes involved, some contaminants would be excluded, and so they would be kind of more retarded as they would go through, and that would make it so that they're even slower. So let's just jump into some fun data here. So what we've done in our lab is actually look at some of the things. So let's look at time and concentration in some hydroponic systems, right? So these are plants grown in hydroponic, and we're looking at benzoisothiazole. This is a compound, it's antimicrobial that's used ubiquitously in consumer products as well as in buildings, and so it's on the outside to inhibit mold growth and mildew. And so it's commonly found in stormwater. And what we're finding is that we can, plants can rapidly take up this compound, right? This is hours, not days, right? And we can repeatedly expose this, and the plants will rapidly take up this compound. And this is very much a relevant time scale for what would be happening in stormwater. Nothing's happening in our abiotic system or something. So this is just plant impacts right here. And so we've done other studies with other compounds, and one of the things that we can do is we see is that if we look at just the kind of mass balance and kinetics here, is that idea that the maximum amount of uptake would actually be the water is not necessarily true for some types of emerging contaminants. And in fact, for some things like this is an anticorrosive, benzotriazole, you'll see here, is that the amount of mass taken up by the plant in the same amount of time is actually 15 times more, and some are even higher than that, for the amount of water that's being taken up. So we also have some other evidence that it follows classical enzyme kinetic models and things like that, which is really cool, suggesting that these are actually active uptake processes. And there's all kinds of cool literature in the plant literature about why the plant may do this, but there are basically non-specific transporters that are trying to scrounge for these types of small molecules, that the plant would want, give it advantages for growth, but they are very much look like these emerging contaminants. So one of the things that we did here was we actually used a base molecule for a fungicide, which is found all over the place. And what we did was we were able to go in and order all kinds of different variations on it with different properties in its functional groups and different positions. And so it's a benzimidazole fungicide. And one of the things that we did, and Danielle here was actually involved with a little bit of this work, and it is actually going in and looking at the uptake kinetics associated with the different properties and position effects. And one of the key take-homes here is that properties make a big difference. For example, a highly electron withdrawing group, such as the cyano group, makes it so that the uptake kinetics are very, very fast. If we go and we look at the positional effects that also can change the kinetics, one of the key things here is this ability to have a rapid conjugation reaction with this unbonded nitrogen group right here. And so this appears to make it so that we're able to go in and the plant takes it up and transforms it quickly. So this was a fun study, and it's really fun to be able to go in and instead of taking a compound by compound approach, be able to use things like computational chemistry to predict some of the things, because you could measure compounds forever, but because there's always more chemicals to work with. But how do we make predictive forward predictions? So what does the plant do? What does the plant do with it once it's inside, right? It's, some things are just there, but one of the things we did was we actually looked at what it's doing. Let's play with benzotriazole, one of our favorite chemicals to work with. One of the pathways that we saw is a glycosylation reaction. So it's a conjugation, right? It's adding something to the molecule. In this case, it's a sugar. And what that means is this is exactly what's happening in our liver. What we do is we detoxify things, adds some sort of sugar onto it to make it more polar and more easy to excrete in our kidneys, right? So this is the same thing that the plant is doing. And basically up until this time, people thought, okay, great, it would just go into a cell wall vacuole or something like that into the plant. But we've actually seen evidence that some of these things are leaking out of the plant or actually being excreted in the same way that happens too. Another pathway is conjugation with various different amino acids, right? And what's really cool about this is that we actually see for each one of these, right? You could see the parent compound is in blue, and then you see an analog in here, but instead of the benzotriazole contaminant, we see a natural indole ring. And for each one of these things, there's an analog. And this top one, all the way out to the right there, is tryptophan, right? That's the thing that your grandpa told you in Turkey made you sleepy at Thanksgiving, right? Not totally true, but whatever. It's an essential amino acid. It builds proteins, really, really important. The bottom one, these are actually the storage forms of auxin, right? A plant hormone signaling that impacts growth and development. So what this is telling us is, and we're continuing to explore some things in the metabolomic world with these, is that these things are being taken up into the plant and actually impacting the growth and development potential for the plants too. So are these things relevant in stormwater, right? We can do a hydroponic study, but we actually collaborated with a group in Toronto, and they were doing a dosing study of benzotriazole into a bioretention cell in Canada. And so they were mostly using it as a tracer, right? It's not supposed to really react or anything like that. It's pretty microbially recalcitrant, which is why it gets exported from wastewater plants. One of the things was that because we had synthesized these authentic standards of plant metabolites, we could actually see that some of these things were actually being exported through the underdrain of their bioretention cell from plant metabolism. So this is definitely relevant as far as understanding what is going on here in terms of being taken up into the plant, transforming, and then potentially being exported as a form that we don't necessarily know what it is, right? Which is sometimes referred to as a phenomenon of metabolite masking, because if you're only looking for the parent compound, you're not going to see this metabolite using conventional analytical techniques. So it is stormwater relevant. Even, we can scale this, do these mechanistic studies in the lab, and then we know that it's actually relevant. So let's shift gears here a little. Another, I like to say, neglected biological process that could be very relevant in bioretention systems is the use of fungi, right? Why would we be interested in fungi? Well, for some of these compounds that tend to be a little bit more biologically recalcitrant, right? Harder to break down. Fungi have evolved over the course of millions of years to break down things like lignin, wood rot fungi they release strong extracellular enzymes, right? All of us have cytochrome P450 enzymes, right, in our livers. Bacteria, plants, fungi, right? And those tend to be very specific, right? Enzyme substrate binding, right? These types of extracellular enzymes are kind of like shot out to pre-digest some of the things of their wood substrate, and then the fungi can break them down. So being non-specific and very strong has advantages for contaminants that doesn't require the organism to kind of adapt or anything. So our question here is, what are the impacts of fungi on trace organic contaminant fate in stormwater? So as you can kind of tell from our modus operandi here, we like to start out with a batch system, right? Sometimes we work with field, we work with batch, right? But here we do like a fungal treatment and a no fungus control. So this is just C over C naught type of thing through time. We looked at a couple different types of tire wear compounds. And you'll note we were doing this work like when some of the researchers were doing things with thyroid compounds before 6-BBDQ was discovered, but these are some of the things that were presumed to be high impact. So we were looking at HMMM, acetanilide, and diphenyl guanidine. You'll see that some things fungi didn't touch. Some were broken down pretty well. Obviously, the kinetics are much slower, but this is something that seemed to work. So I'm going to take one method detail because this is something that I think is important. We're using a cool cutting edge technique with high-res Orbitrap mass spec. And the way we do this, whether it's plants or fungi, is we could get lots of biological replicates and isolate the mechanism. And what we can do is sometimes do library matching, go and say, okay, this is this peak, what's going on here? And then filter our, say, 10 to 20,000 data points for a single sample, and then go in and do fragment matching, which is kind of a break the molecule into pieces, Humpty Dumpty it back together again, and see if there's something that either matches a known pattern or is a novel transformation product. This is a really powerful tool for discovering new things. So one of the things that we did was we actually figured out what this pathway is for HMMM. It kind of looks like a spider, and the fungal enzymes are able to go in and kind of chew on it, break it down. And I'll highlight here that one of the end products is melamine, which maybe you've heard about that in dog food and other, like, it's not necessarily a benign thing either. So one of the take homes here is that it's really important to understand these products and pathways, right? We know that what we discover in the lab can be very relevant for the field, but byproducts are not necessarily benign either, right? What it started out with is not good, but sometimes, most of the time, biotransformation yields less toxic things. Sometimes that's not always the case. We don't know for sure whether the fungus, because with this analytical technique, we couldn't tell whether it was breaking open the melamine and degrading that. So that's still a possibility. We wanted to see whether this is something that happens in real life in the field. So we were working with a local community where they have a lot of green infrastructure, a suburb of Iowa City. And so we went out and we were gathering these soil samples, and we were saying, well, is this something that happens already? Or is there an opportunity to potentially bio-augment bioretention cells with fungi? So this is actually, we did a sequencing analysis here of the fungal communities in these systems. We looked for biodegradation functional genes associated with fungi, meaning like, are the enzymes there potentially associated with fungi, which is cool. It tells you whether they're there. We also looked at some like big family-based things in here. This doesn't tell you necessarily whether they're specifically biodegraders, but this Biceta macota is the class that include brown and white rot fungi, meaning that there's the potential there. It also means that there's the potential that maybe we could improve the system, tweak it a little bit, or make it so that we could have, bio-augment it and make fungi a more important part of the process. So let's get back to our original question. Can biological processes remove contaminants? I think, yeah, I think we know that this is an important thing. It's one of the things that we want green infrastructure to be green, and we want these things to be low maintenance and self-sustaining to a large degree. And so this is a really important process, right? We don't want to just capture the contaminants, we want to degrade them. We know that there's multiple tools to do this, right? Bacteria, plants, fungi. So that brings us to our next question here, is can we improve our geomedia that's used to go in and biodegrade contaminants by better synergizing sorption and biodegradation? That's really our goal here, right? So one of the things that sometimes limits it is that you've got a contaminant over here, you've got a degrader over here. How are we going to get them together? We need a little bit of matchmaker here. So let's get back to our original challenge, right? We want to have a system that maintains rapid hydraulic conductivity. That's one of our design goals. We want to have infiltration and keep it fast, right? We don't want, one of the ways that we could make it so that we're going in and sorbing more is by increasing the organic matter content in there. We also don't know that loading a bunch of compost into a bioretention cell is a good recipe for like nutrient leaching, right? So one of the things that people are doing is going in and doing things with adding black carbon like biochar into these systems to try to like this is not going to leach. It can make like nutrients. It can make it so that you're maintaining hydraulic conductivity and everything. And we want to make it so that you can better adsorb contaminants. This isn't necessarily new, but using it in bioretention is a really cool approach. What we wanted to do was to make it so that we're also, biofilms are going to grow on these, but how can we make it so that we're going to go in and bring them together and bring them together faster and potentially use this as a tool for bioaugmentation too. So we we developed something that after a long time and lots of trial and error, and we don't normally work in the field of materials development and stuff, is what I call biosorb beads to help deliver contaminant degrading organisms, so a bioaugmentation, and with the goal of capturing contaminants with sorptive materials and then bring degrading organisms into the same proximity as the contaminants so it can help break them down more. So the goal here, right, in wastewater treatment was revolutionized with the activated sludge process by going and decoupling the hydraulic residence time, which has to be short, from the solids residence time, which can be much longer. So our goal here was basically kind of the same thing. We want to go in and decouple the rapid short hydraulic residence time, maintain hydraulic conductivity, while also integrating the longer necessary chemical residence time to allow for biodegradation to occur in situ. So we know that biofilms can regenerate these things or regenerate the sorption capacity in situ. We collaborated with some folks on work in there. And I'm going to kind of go through a little bit of how we built some of these things. We used a natural polymer, alginate, which is sometimes used in drug delivery and all kinds of other types of things. We used recycled materials from a lot of different things. Iron-based water treatment residual was something that normally the drinking water treatment plant would have to pay to dispose of. And that was added for iron for phosphorus removal. Inspiration here, right? And using powdered activated carbon, we were just using some sort of powdered black carbon. We were trying to do this this was a direct follow-up from our fungi work. So we wanted to see, could we optimize this for fungal bioaugmentation? So we added wood flour, which is a byproduct from flooring manufacturing, from drum sanders, from hardwood floors. It's sanding dust, right? I made some in my garage, right? Then we had fungal liquid culture, which was used in, and that was added into there too. We can put this in. We also added a known electron shuttle for potentially improving the biodegradation potential. Added that in. It goes dropwise through a peristaltic pump into some sort of either di or trivalent cation solution and then it instantaneously forms a hydrogel. These dry on wax paper, so very high tech, right? And then they dry down about half their size to kind of like, they look kind of like shotgun pellets, right? They're just little beads. And I actually have some here if you want to do show and tell. So you can touch them. These are abiotic, so there's nothing here nothing encapsulated here. But the way that this looks is actually pretty simple. You mix everything together and it goes in and we put it on there and they kind of look like bullfrog eggs when they first go in. And it's an instantaneous process. So it's pretty cool. And they dry down and you can then store things for a really long time in there. So we did all kinds of things where, because you can make lots of variations on the theme, we could go in and do things. I'm not going to get into the details here, but by testing all these different variations, right? What if we vary the crosslinker? What if we added different alginate concentrations? How does that affect the surface area? What are we doing here? And I'll just give you a couple snippets here. One is that by adding in these things like the iron-based water treatment residual, it goes in and it substantially increases the strength of the beads, right? That's important because we don't want them to just crush. One of my big concerns is that, the way that the hydrogel structure goes with this kind of, they call it an egg box gel structure, is that potentially you could get a flush of salt coming through your system, and then the whole thing would just dissolve by ion exchange, right? So we kept beads for a year in synthetic seawater, and they do swell, but they didn't actually fall apart. So, I mean, it's all natural materials. They're not going to go in and last forever. You don't necessarily want them to be there, but it's not something that is going to go in and, like, if they break down, not a big deal, right? You've just added some stuff into there. So even after keeping things in there at room temperature for like a year, we can go in and add these beads into some nutrient media, and then we get fungal hyphae growing right back out of it. So it's a good way to keep them for an extended period of time and add that in. So this paper was published and on kind of the development aspect of it. We did a bunch of testing, and we just did several different things, phosphates, some neonics, which are insecticides, mostly because the alginate changes some of the charge distribution effects of the black carbon, so we're kind of probing some of that. We looked at a couple different PFAS and a tire compound, mostly, right, something like phosphate and the PFAS are only going to be removed via sorption, right? We don't expect any biodegradation of PFAS. That's not going to happen, right? Just a couple of things, hitting a couple highlights here. Just from sorption, we know that, for example, that the iron-based beads that are cross-linked with iron and have iron water treatment residual remove more phosphorus. Not earth-shaking, makes sense, but that's really cool. They change the bead structure, right? They look kind of like a red blood cell. They're kind of spheroidal, flattened disc-shaped, whereas the calcium ones are like a little sphere perfectly round. We found out that charge distribution of the molecule does impact its sorption capacity. It kind of reverses some of the tendencies associated with conventional black carbon. So there's lots of details here, and you're welcome to look into I'm happy to share any of the papers with people too. One of the big things was also that we looked at combined sorption and biodegradation, right? That's the whole point here, using fungi and acetanilide as a tire compound. One of the things that I want to point out here is that when we did two different, like trying to kill the fungi or inhibit them, one with autoclaving, and so the red line should be considered like just sorption because the fungi should be inactivated. The other we also did with, we inhibited them with azide, which normally kills fungi. It's a chemical treatment. But what we found was that it didn't kill them. They were happy to hang out inside the beads. And so this was kind of ruined our experiment, but also told us something really, really important, that encapsulation protects these microbes from really adverse environmental conditions. So we don't normally do stuff in materials development and stuff like that, but we were convinced to go down some of the IP development type of stuff and whatnot, mostly because it could be a way that people could get this out into the world. So our newest thing here, because again, this is kind of a platform technology, the grad student was like, what if we try to look at nitrate? Because nitrate is something that can be biotransformed. It's also really soluble. So we built some beads that had anti-exchange resin in it. Pretty cool, right? And that's really basically the big thing. You can see the red line here is just the removal due to abiotic processes, anti-exchange. And that would be considered like just sorption onto there. If we go in and look at sorption and biodegradation, this is very preliminary data. It's not yet published or anything. We're trying to just like see if this can go someplace. And like, I think we can probably do better with different types of anti-exchange media, but it's promising that we're getting removal, including even where we start out the beads in bulk aerobic conditions. And inside the bead, it will maintain anaerobic conditions, we think, and they're removing the nitrate, which is pretty cool because potentially you could make it so that you could take the beads and do like what we're trying to do here. We did our first field deployment. We scratched away the mulch and then just integrated it in with a tine hoe, right? So this would be post-construction amendment. Potentially, you could get denitrification by even in a bulk aerobic condition. So you wouldn't have to totally design an under drain or subsurface saturation system because maybe you could get removal during the infiltration and then the microbes would keep it up. So can we improve Geomedia for capture? I think, yes, we can do that. And by like making better media to improve the capture and synergize removal. And my last and kind of short thing, which is really kind of work in progress here, but I really want to share it because I think it's a really cool opportunity for new directions, is to think about new measurement approaches for understanding these complex mixtures, this chemical soup that's here. So this is the work of Stan Coles here. And what we're doing here is a really cool field-based project where we're going in and there's a community where they have a lot of green infrastructure, same side of the street, or different sides of the street, same street, where we can kind of use treatment and control in here. And so he's doing some first flush samplers in the inlet and the outlet of a bioretention cell. And we're doing non-target high-res mass spec analysis. And Stan is also from Minnesota, so he likes to use a fishing analogy for what this is. And right, so targeted analysis is where you're just kind of going in and casting and looking for one contaminant at a time. This non-target is you're going and throwing in a whole seine net and trying to see capture everything to understand what's in your system. So you get like, this is a simplified version with just 2 samples. You end up getting like gigabytes of data for each sample and 10,000 data points and all kinds of crazy stuff. But this is a good walkthrough. So we saw like in our field sample, we've got an in and an out. And so we can, the columns here in these heat maps tell you, okay, what's in the influent, what's in the effluent, what's going into the bioretention cell, what's coming out. And the color here, the blue, means that there's greater relative representation, which means that there's more of that compound in that particular, in the sample relative to the other, right? And so this, right, if we have got blue in the outflow, that means it's greater in the outflow. That means that it's a net export from the system. So one of the things that we can see here is that for the majority of features, right, the thing on the left, that's a hierarchical cluster analysis. It's kind of like a family tree of chemicals and how related they are. We don't necessarily know what all of them are, but we can see that most of the features are being retained rather than being exported. There could be things that are like, we found plant root like hormones in some of the exports, so it could be totally natural. We can also go in and look for things that we weren't looking for. What does that mean? Well, if there are things with spectral libraries, we can see, for example, flame retardants being exported here. It probably was from a prior storm, captured, and then passed along. Where we can see that atrazine, for example, is retained. So just preliminary data, right? So what we can do here is now look at big total mixtures here and see things that are chemical trends. For example, in these preliminary results, we can go and we can see that the compounds that are retained tend to be higher molecular weight, which makes sense, right? Bigger things are being stuck more, and those that are more apolar, right? And that makes sense too, those that are more hydrophobic. But now we can look at this in kind of the totality of that mixture. So this, I think, is a really cool emerging measurement approach that we're going in and getting to put in and pull into place here. So we're excited to, like, we've collected a year's worth of data. So now it is taken like a week for the computer to just keep running on crunching the data for phase 1. So it's a big deal. And we hope to be able to evaluate that for the efficacy for these complex mixtures. So I want to kind of close here with what I like to think of as a new framework for bioretention, right? Can we couple rapid contaminant removal via sorption with sustained biological process to remove contaminants in situ and basically make systems better for emerging contaminants, but that are, for all kinds of good, important reasons, but are also helping us make it lower maintenance too. We recently put together an editorial piece with Steffi Spar at the Leibniz Institute in Germany and then Elodie Passport at Rutgers. And what we put together here was what we called a tiered complexity framework for treating water-soluble hydrophilic contaminants in green stormwater infrastructure. I put that QR code on there because it's just online now and it's a free open access and stuff if you want to look at that. But basically what we're trying to do here is look at what can we do? We know that there are limited resources in the world for implementation. what can we do to make it so that we treat the most water quality improvement with the least amount of effort and resources? And so what we're doing here is looking at kind of a tier 1 intervention, which was to go in and improve the system hydraulics, because sometimes you don't even get complete water spread over the entire system, and sometimes there's not adequate contact time, right? Sometimes the systems are just simply poorly designed or something like that. We called tier 2 was improving the media. This is more expensive, but we can, this is very doable, right? If we can advance media and make it so that you can sometimes either post construction or use different layers of media, and this could greatly improve contaminant removal. Then what we called tier 3 was more advanced treatment options, things like more advanced oxidation removal and stuff like that, which would really be only reserved for contaminant hotspots, certain types of industrial stormwater, places that are highly ecologically sensitive or impacting drinking water sources, because it's really energy intensive and expensive, right? So thinking about that in a framework of like, what do we need to do so that our watershed can maximize the implementation of our resources? So with that, I am very thankful for all the people that have worked on these projects throughout the time here and stuff like that. And I am happy to take questions.
Wonderful. Thank you, Greg. All right, we will take questions from our in-person audience and our online audience. Would anyone like to get us started with questions for Greg? All right.
First. Wow. So I got 2 questions for you, if I may.
All right.
First is, you were talking about plant uptake in the context of rain gardens. Question would be, how important is harvesting the plants at the end of the season? If they're allowed to decompose, does that just release what's been taken up? The other question is, it looked like most of your work is with rain gardens. I'm interested in the extent whether this would work better in systems that are based on mature trees. Spectacular biological community in the root zone volume.
Sure.
And perhaps greater plant uptake and more stable and durable plant uptake as well.
Sure, yeah. I mean, part of it is just the framing of the problem, right? I mean, classical phytoremediation of going in and doing like a contaminated solvent plume or something like that is very, like, they're using phytophytic trees and stuff like that because they're big and they pump a lot of water and stuff. So I was trying to frame it from a stormwater perspective, and usually you don't have a lot of trees like that. But I think that it's, these are important processes to understand, but they can also be like, they can be really relevant even when we would expect them not to be. To go back to the first question about the, would it be important to potentially consider the byproducts and stuff like that? It is really interesting, right? Because they can sometimes be present as residual. And one of the things that we are interested in, we actually just published a paper a couple of weeks ago about, from benzotriazole is actually found, for example, in recycled irrigation water. And so like from a crop exposure perspective and stuff like that, and following the transformation products through that and everything. And this is something that it's like, because plant conjugation, the core structure of that contaminant is unlike, for example, the fungi or the bacteria that breaks it apart. The plants usually kind of add stuff to it. Sometimes it gets basically irreversibly bound into the cells and stuff, but sometimes for these more polar ones, it's not, right? So it would be something potentially to consider, but a lot of them, I wouldn't be overly concerned with a lot of these. The thing that sometimes would be more concerning to me would be like metals, because those are not gonna eventually really, I mean, those are build up and accumulate, no different than they would in your media.
Very good. Let's take an online question.
Do you know how many biosorb beads you would need per square meter in a bioretention facility, and how much would these cost if mass produced?
That's an excellent question. And unfortunately, a lot of funding agencies are really great for going in and making things on a palm scale, but not on the scale up from their scale. So like one of the reasons why we actually did go down the like product development IP route was because it actually lowers some of the barriers to implementation. If anybody is interested from like building these things, I'm not in it for making money. I want people to implement these things. So if you want to talk to the university's like licensing thing and make it, awesome. I'd love you to do it. It's like, it's very scalable, right? You could totally do it. We have not. It took, I mean, I had an undergrad making these, she would make them before class each morning and just make them a little bit at a time. And like, it took a long time to even make like a gallon of these just to put into one. That's probably the bare minimum of what you would kind of need. You probably want a lot more. I don't have an exact number. You probably would estimate it based on like what would be some of the sorption capacity associated with different things in there. So I don't know that off the top of my head, but it would be like consideration number one in scaling.
Next question in person, right here.
So Greg, you said some of these conjugated forms might get bound up in the plant cells, and you also talked about masking. Could you have a situation where the plant conjugates it, excretes it, and then something else deconjugates it, and you just transport it down at a different, a more complicated pathway?
Yes. We actually have a project that we're doing that exact question right now. So the answer is, it depends on what form it is. So long story short is, and I guess I can say this, sure, is that like the bacteria will deconjugate some forms, but not others. And sometimes they will turn, like the bacteria will actually kind of, like they're gonna attempt to detoxify it. Some of them will go in and try to eat it as a kind of a food source. And the sugar conjugated ones are much more attractive for that. But it does fundamentally change the properties of it. Like it's gonna be more polar, it's gonna move and stuff. And so it's, we actually, in this latest paper, we also did some like toxicity computational modeling of that. So we could see that like between parent plant conjugated metabolite versus like what the digested metabolite is, the toxicity profiles change as it moves along through that pathway too. So it's like, it's important to understand these things, but this phenomenon of contaminant masking, where it's like, if you're only looking for what you're looking for, you're going to miss that product in the environment.
Wonderful. Let's take another online question.
Is there any potential for the biosort beads to treat DDT, DDD, DDE, or chloridane? Could they pull these constituents from the soil to retain them?
Totally. You probably would see like, because DDT and DDT metabolites are so hydrophobic, they would probably already, if they're in stormwater, which is not going to be often, but they would most likely already be particle associated with something else because they've got a log KOW of like 6, right? They're going to be stuck onto something else and probably be filtered out more so. But I mean, the field of using geosequestration and geosorbents for contaminated sediment remediation for things like DDT and other legacy contaminants is well developed. And the focus on that is mostly just on sorbing things and causing irreversibly bound sorption. It's certainly, it would work. I don't think you'd probably, because they're pretty biologically recalcitrant, it would probably be slow to break down like anything else there.
Next question for Greg. Oh, I got one.
Thanks, Greg. A two-part question.
Right.
Sorry if I missed this. One would, going back to additives to soil, let's say for bioretention, one would argue that there's natural fungi in the soil.
Yeah.
And this work would add it.
Right.
What's the difference in those?
Sure.
Why wouldn't the natural fungi just populate and do this? And then second part question, whether we add, well, let's say you add through the stuff you're doing, do we have any evidence that that fungi will repopulate and grow without us with further additions?
Sure, yeah. So, with any type of bioremediation system, oftentimes there are native organisms there. And so you can change the environment to allow them to grow, but sometimes bioaugmenting can make the system faster. But sometimes, I mean, the Basidium acota is a really big group, right? Like it includes things like white and brown rot fungi, but that doesn't necessarily mean the organisms that are highly favorable with the strong extracellular enzymes that are likely to break them down aren't going to necessarily be there. And we don't necessarily know the populations and stuff like that. So it seems as though that this is likely something, but it seems like there's bioaugmentation opportunities. We can make, by tuning the biosorb beads, we can actually make the environment more favorable to bacteria or fungi, because by changing the cationic crosslinker, we actually change the internal pH. Fungi like it really acidic versus more neutral for bacteria. The other thing is that we can also put beneficial bacteria in here, or however we want to kind of do it too. So there's different opportunities. We were kind of using this as an outgrowth of the fungi too.
Wonderful. Let's take another online question.
What is the lifespan of these beads if studied? Do they eventually have an end-of-life stage and need to be properly disposed of?
Yeah, that's an excellent question. In the field, I don't know how long they would live as far as there being an intact bead or not. I mean, there's some evidence in the literature that because they're a natural polymer that they would eventually break down. But if anybody does microbiology type of stuff, right, just a plain agar plate, bacteria don't really grow on that, right? Unless there's some sort of really specialized thing, right? You have to put a nutrient media in there to have them do it. Alginate isn't a super favorable food for most microbes. So they would probably last for a pretty long time, but they're not gonna last forever. There's other types of things that could, but if they broke down, they're not going to break down into something toxic. You could use, for example, aluminum as a trivalent crosslinker. Would that be good? No, right? We don't want to load a whole bunch of aluminum or zinc or something, all these things that we're trying to remove into a bioretention thing. Just because we can doesn't mean we should. Does that mean that obviously anything that had adsorbed onto the media but wasn't degraded is still going to be there, right? This is mass balance 101, right? So if you've got sorbed phosphorus onto the water treatment residual, it's still going to be there. If you have PFAS that's on the black carbon, it's still going to be there. So it's not going to totally change that, but it's going to prevent those water-soluble contaminants that normally would pass through either into your surface water or into your groundwater from, it's going to stay more into the bioretention cell. So yeah, there's still some maintenance that is involved, right? I don't know necessarily how long they would last, but I would expect it to be, we'll see how these are too. So it should be years based on the literature.
Another question. Oh, we've got a couple right here.
Hey, I was wondering, you talked a little bit about being worried about salt impacting the integrity of your beads. I was wondering if you could speak about how seasonal flushes of salt in places like Minnesota and Iowa might impact the performance of these GSI when it comes to these biological processes.
That's a really, really good question, because, and I think it's something, one of the things that we kind of closed our tiered framework editorial with is that there are some things like road salt, like trifluoroacetic acid, that are never going to be effectively removed in bioretention. So the only feasible approach is going to be source control. I think there are multiple things, and we were concerned about the structural elements of the beads falling apart. It didn't seem to be as big of a concern as I was maybe making it out to be in terms of a worst case scenario. But there's no question that going in and loading a whole bunch of that into a bioretention cell is not going to be good for its biological systems. One of the things that, for example, the Minnesota Pollution Control Agency's planting guide suggests putting in, for example, plants that are pretty salt tolerant because of these fluxes of salt into the system. But it's not good. There's nothing that you're going to be removing with that.
Very good. Let's take another online question. I think we have a few more.
Have you looked at plant uptake of contaminants during both the growing season as well as during senescence? What about reintroduction of contaminants during senescence? Have you looked into plant uptake seasonal?
That's a really good question, right? That's not something that we've directly looked in, and that kind of relates a little bit to the prior question as far as like where would it go and everything. There are some some short chain PFAS will be taken up by plants, right? Like nothing's gonna really happen to them. And so it depends on how it's going to be biotransformed, biodegraded. Particularly with these types of plants, I mean, trees do have some degree of still movement of water and stuff like that, even when they're dormant, whereas most of these other plants are going to be completely dormant. So it's really only going to be a efficacious process during the growing season.
All right, we'll go in person again.
Oh, I had one comment about the 2 questions ago about the salts. We did find from some of Kim Palce's work that the seasonal salt flushes did actually remove or mobilize some previously deposited metals.
Yeah.
So presumably the metals were captured via cation exchange, and then high salt flushes were able to push them off the material. So that was one effect of salts. But
Thanks for that.
my question, Greg, was, great job, by the way, was about the high-res mass spec non-target. Normally, I don't think of that as being quantitative because obviously you don't know what you're looking for, so you don't have standards.
Right.
But obviously, if you're comparing in and out, you're just doing
Yes,
peak
it's
area,
relative, right.
peak areas. But you don't really know, like, I don't know how abundant they are if you want to do like toxicity or something like that.
No.
So I don't know, could you talk about the quantitative aspects of that or lack thereof?
Yeah, so these are different tools for different questions, really. And I mean, we can use some of the same tools, like using the same instrument, depending on how you design your experiment and what you're setting this up for. Like we can use the high-res mass spec for doing our products and pathways analysis to try and predict what's going on. We can do it for suspect screening to see like, okay, does this match a database from NIST or something? Like, does it match in here? When we're doing kind of these types of things where it's just looking at the mixtures, you're looking at mixture dynamics, and it is relative to some other condition. And so, yeah, it's peak area relative to for its own self. Sometimes if we go in and put, like we've created some standard mixtures, so you'll know what is in the standard mixture so that then you can get to the highest level of confidence identification. And it's like, oh, okay, I know that it's more than 10 nanograms per mil or something. You can say whether it's more or less, but you don't, it is not quantitative. It's an exploratory thing to look for like, okay, we can identify it. We can identify it to a certain degree of confidence, but then it also can help inform the next thing, like, oh, well, how good is this for retaining these types of things? And if you want to do quantitative things, then you have to use a different tool set, which is, they're all complementary. We have to use them all. But this is something that's a newish thing that is, I think, really important and not really applied much in stormwater.
Very good. Well, we are out of time for the Q&A right now. We're going to take a quick break and come back for our panel discussion. So please stay tuned. We'll start again in 5 minutes. Thank you so much. Welcome back to the Minnesota Stormwater Seminar Series. Again, my name is Andy Erickson. I'm super excited to be hosting and moderating our panel discussion on this topic today. I will start by introducing our panelists, ask for their relation to the topic, and then we'll dive into your questions. Again, if you're in the room, just raise your hand. You can ask your question. If you're online, put your questions in the Q&A box, and we will go back and forth between our in-person and online audience. I do want to mention before we start the panel discussion that this seminar series is also co-sponsored in kind through the Cold Climate Stormwater Center of Excellence, which is funded through the U.S. EPA and in partnership with the University of New Hampshire and the National Municipal Stormwater Alliance. So we're super excited to be able to bring these topics also through the Cold Climate Stormwater Center of Excellence. But without further ado, I'm going to start introducing our panelists. Our first panelist on my right, your left, at the far end of the table, Dr. Bill Arnold is a distinguished McKnight University professor and the Joseph T. and Rose S. Ling professor in the Department of Civil, Environmental, and Geoengineering here at the University of Minnesota. Bill, from your perspective and experience, how do you relate to the topic today?
So I work with Andy as part of it. And we've got projects that are looking at doing surveys for detecting various tire-derived and other stormwater-related chemicals in the Twin Cities and other areas of Minnesota, and looking at some techniques for their removal.
Very good. Thank you, Bill. Our next panelist is Justine Dauphinais, who is the Water Quality Coordinator at the Coon Creek Watershed District. Justine, from your experience and perspective, how do you relate to our topic?
Yes, thanks, Andy, and good morning, everyone. So I relate to this topic not as a subject matter expert. In fact, I struggled my way through organic chemistry, like most of us in this room probably. So I come at this more from a urban stormwater practitioner, that this is just one more thing I'm worried about that keeps me up at night, of what is in my stormwater being transported to our receiving waters. In the Coon Creek Watershed District, we're located in the North Metro on a glacial sand plain, and our stormwater is drinking water, both from groundwater sources and because we drain to the Mississippi River immediately upstream of the intakes for the city of Minneapolis and St. Paul. So I think we're kind of uniquely located in a place where getting a handle on these contaminants that might have direct impacts to human health, as well as impacts on our biota and our impaired streams, is important.
Very good. Thank you, Justine. And our next panelist is Richard Kiesling, formerly of the U.S. Geological Survey. Richard, from your experience and perspective, how do you relate?
Well, we've been looking at potential removal or amelioration of CECs through in stormwater through different kind of biofilter biomedia, looking at influent effluent concentration and occurrence differences. And I think what we've seen today is that we need to have a better understanding of mechanism, and not only mechanism, but the timeline for sequestration and eventual fate. And I will say that when my colleagues and I give presentations on the work we've done to date, we almost always get questions about the fate of these compounds and what's really happening to them. And I think Greg's presentation today has given us a lot to think about and talk about.
Very good. Thank you, Richard. I won't ask for Greg's relation. He gave the keynote and kind of set the stage for us today. I'm going to start our panel discussion. We heard from Greg about some of the new exciting things that are happening, about what we know and maybe some of the things we don't know yet right now. I'd love to hear from our panelists. What do you feel are the biggest challenges and hurdles related to these chemicals and stormwater management in today's industry? And at this point, anybody can chime in.
I'm going to jump in first so I can take the easy answer. So from my perspective as a practitioner, one of the biggest challenges is that most of these things are unregulated. We do not have state and federal mandates to look for these things and to address them in urban stormwater. So it's something that, working for a watershed district, this is just, it's a secondary benefit, that we're looking at as we try to characterize and treat our conventional pollutions. CECs is kind of an added bonus.
Other thoughts?
Well, I think another challenge is just the sheer number of compounds, right? And if you digest the tire, there are thousands of chemicals in that tire, and they're often proprietary in how they're formulated too, right? And so just trying to figure out what's there, what might be toxic, what's the most abundant, and whether that's the most toxic or something else, that's all very difficult. And then the range of chemical properties varies so much in their degradability that it's a difficult task.
I think I would add to that the ubiquitous nature of these CECs is something that's been striking in the research results, especially as it relates to stormwater, with stormwater profiles that often look a little bit like effluent profiles from point source treatment. So my coming to the issue of how these are delivered to the nearshore environments and lakes, for example, and the biological impacts goes back to the question of which compounds and what mixtures and to what degree are they having a biological impact. And I think the focus on mechanism, on removal, and removal dynamics might be very important, but I think the idea of a risk-based approach to the multiple compounds that we see may give us some hope in how we might treat them.
I was going to say also and Greg alluded to this too, right? You have chemicals you've got salts, you've got things from tires, you've got pesticides, you've got PFAS, you get pharmaceuticals because you get leaking stormwater or wastewater infrastructure, right? It's just the mixture of stuff and not things that we necessarily see together in other scenarios. You see them in stormwater.
I'd add too that, like, and this is just an inherent challenge associated with stormwater, but the concentrations are relatively low, but from a mass flux perspective in a watershed, they're very high. So it makes economic treatment not something that's very feasible, right? And when there is not a regulatory driver, particularly for emerging contaminants, you can have really important impacts on watersheds that are not necessarily always apparent. I mean, sometimes, like, we were talking at dinner last night about, well, how do we even know what an emerging contaminant is or when it's emerged? It's not like a mayfly that comes out of the water, right? That's emerged. But much like a mayfly, they can spend a lot of time in the water. Nobody sees or appreciates they are except for the fish that are eating them.
And I think too, you know, not only the number of chemicals and the diversity of properties of these chemicals, but then the cost to even look for them, I think, is also a major challenge because there's so many chemicals and there's sometimes few laboratories that can actually do the analysis on some of them, that the cost to try to look for them even can be pretty expensive and cost prohibitive. So let's dive into your questions. What questions do you have for our panel discussion today? I know we have several online. Why don't we take one online while the in-person audience thinks?
What do you think about studying these processes that Greg presented on in highway roadside ditches, which have been shown to infiltrate a lot of the runoff from the roadway, even though these ditches are not usually engineered for infiltration?
I think that's a really good and cool opportunity. And I know there are some places, actually, one of my current students did her master's at University of Delaware, where they were adding in biochar into roadsides as a way of going in and using these. This is not necessarily stormwater, but one of the ways that people were in Iowa are looking at roadside ditches as a way of improving the denitrification potential in there. So trying to think of, it's a public right of way, right? And it's maybe small in any one place, but linearly, that's a lot of area. So I think it's a really cool thing, and scaling these things up. I mean, my postdoc advisor always talks about the 3 M's of environmental engineering, right? You want to do mechanistic studies in the lab to try and understand these processes so that you can gain generalizable insights, but then you need to go measure stuff out into the field, right? Those are complementary approaches, and you have to have both to make sure that it's relevant, and then create models so that then you can have forward predictive things, so that put those all together, and then this is how we actually understand systems. So scaling is really important.
Other thoughts? Highway runoff.
I guess I'll chime in that I hadn't thought a lot about, roadway ditches until this past water resource conference at the U of M, where there were like a dozen topics on like soil health and roadway ditches and just how important it is to think of these not as, compacted soil with turf grass, but as part of a stormwater treatment, that we need to make sure the soil's not compacted. We can add additives like biochar. We can plant diverse native plants. And I think all of that supports kind of the microbial and fungal communities, and kind of more variety is better, essentially.
It's actually been, I'm not sure exactly what the designation of roadway that they use, but in Iowa, it's DOT regulation that state roads have to actually have native prairie planted into the right of way, which is really cool because it has a lot of these benefits. It reduces maintenance requirements, but also this is a really important pollinator zone too. And actually roadway right of ways are the single largest form of public land in the state, which is kind of depressing, but it's in a lot of agricultural states, it's not just there, but there's a lot of places where that is true.
Other thoughts on highway runoff? I think something to think about related to one of the questions asked earlier is, what happens with this after its life is used? If these chemicals build up in those soils, especially in roadways, we see highway and roadway reconstructions all the time, and it doesn't really go anywhere else. So is that just going to create some pretty contaminated soils? How long would that take? Is that true or not, or is it just pass through? Good questions. Do we have an in-person question for our panel discussion today?
So I understand why the wood decomposing fungi are important because they break down the really tough molecules. But do you know if all of the brown and white rot fungal species would work? Or are there specific species that you think may work better than others? And then my follow-up question to that is, how do you source your fungal cultures?
That's a really good question. And when I put up that big giant little family of, these are Bicedum coda, well, the brown and white rot fungi are one piece of that, right? That's just implicating that there's the potential for these to exist in there. But yes, there are lots of different specific ones, and they will have different extracellular enzymes that can, they can be catalyzed by different metal cofactors and other things like that in there. So they might have different enzymes classes, and some of them might have multiple and stuff like that. So there are definitely different varieties of species. Some of the things that we were using in the lab were actually not something that we would necessarily want to, it's a lab strain that we didn't want to put out into the environment. The USDA actually regulates it even as an invasive species, even though it's something that is common out in the woods. But when we field deployed them, we actually use oyster mushrooms, which are things that you might buy in the grocery store, and they still have some very powerful extracellular enzymes. If we would see in the bioretention cell, we wouldn't actually expect to see the fruiting body of a mushroom in there. Most of it is going to be the mycelium that just are going to be inside the bead and then also move throughout it. And that's like 95% of the biomass of it. You only get the fruiting body if they're going to go out and start to sporulate.
I don't know if anybody else has comments or experience with fungi. That was in person. Let's take an online question.
What do you know about different performance characteristics of different types of plants? For instance, are grasses as effective as shrubs in bioretention?
Sure. Okay, great. So and I'm happy to send that review paper for anybody and stuff. We were really looking at the body of literature on this in terms of specifically bioretention cells, but then also and stormwater systems and stuff, but also plant traits generally for uptake and phytoremediation. And there are some things, in general, we don't usually have shrubs and trees and bioretention cells, at least in terms of small-ish units and stuff. But in terms of some of the root properties, I'm sure everybody in this room has probably seen those pictures of the surface versus subsurface of the prairie roots and stuff like that. Having deep roots are really, really important because it can then create a rhizosphere that's much higher impact within the infiltration zone. It can make it so that water infiltrates there, but that also is a place of nutrient exchange and place for uptake. And then tending to have more fine roots is generally beneficial for uptake rather than just going and having these really deep tap roots, because the surface to volume ratio is different for that. So there are different things that are there. Some of the things that we've used in the lab are we've used model plants that are very standard in laboratory plant fate contaminant work, because it's kind of like the white lab rat of the plant world. And it's, you know the entire genome, and it's very standardized. But we also know from working with other folks that, again, take the system in the lab, understand the mechanisms, and then we actually looked at measuring some of them, the benzotriazole phytometabolites in strawberry and lettuce crops that were irrigated with recycled water in California and Colorado, and you could see some of these things that were higher in those metabolites than they were of the parent compounds in different parts of the plant and stuff. So again, it's like, oh, cool, this is something that is environmentally relevant.
Greg, can I ask you a question?
Go fish.
When you have that that you said that you have that rhizosphere that's, microbially active. How much of what you see in the transformation can you attribute to the plant doing it versus the microbiota associated with the plant?
Yeah, so that's an excellent question. And I think that, as much as we love to do lab-based experiments, right, and we can separate the effects of plants by going in and in those hydroponic stuff, we go in and we actually sterilize the seeds. So they are growing up and there's no bacteria that are in there, and we can decouple that process so that we understand what's going on. But in real life, in a bioretention cell, probably the bacteria that are in the rhizosphere are probably doing as much or way more than what is the plant doing. The plants and the bacteria work with each other. They cultivate each other and stuff like that. So those are really important, and it can help sustain contaminant removal and help sustain those populations. So that's critical. Even though I like to talk about the plants and the fungi because they're kind of neglected, the bacteria are the ones that are doing most of the work.
Superheroes, right? Let's take another question in person. We got one here.
In my previous position when I was working, I had 1 foot in the research world, 1 foot in the practitioner world. And I know practitioners are pulling their hair out, worried about all this stuff that might be coming down the pike in terms, they have limited dollars and what to treat. I'm interested in your thoughts about the communication between the research world and the practitioner. What do we know? What are the most important issues? How do we communicate things when we're confident enough to know something, maybe not everything, but enough to maybe guide the practitioners?
Well, I'll say, one good example of this was the work Ed Kaloje did out in Washington, right? Where they did really elegant detection work to find 6-PBDQ, right? And when they found it and could link it to a toxic effect, that's when you really have to have that good communication and say, okay, we found a problem, this needs to be addressed quickly. The hard thing is, right, the analytical instrumentation has gotten much, much better, right? And so we can see all sorts of different things, and sometimes it's hard to tell what's a problem. And going through and doing the details of mechanism, of toxicity, of loads, gathering as much information as possible to figure out where you need to target your resources is important. And I don't have a good answer, but I think more information is helpful.
Other thoughts on communicating and connecting to practitioners and research?
I think it's essential that it's a two-way street, that talking back and forth, because it's like you want to be doing cutting edge science, and that's what I'm trying to bring here, but I'm also going and recognizing that we want to do things that is this going to be relevant, right? I one time worked during my postdoc project where we were trying to look at all these cool emerging contaminants and this and that, and we were talking to one of the wastewater practitioners, and what are your biggest challenges? And he's like, flushable wipes, right? Like, this is like, okay, we're in a different, right? We got to talk to each other here. Like, okay, what's going on and what are the priorities? And some of that is going and saying like, yeah, what are the things that are potentially coming down the pipeline? But what are some of the things that we should be concerned about? One of the things we talked about a little bit last night at dinner was, are there opportunities where you can get these kind of double benefits associated with it? And this is actually, I'll put a spotlight on Danielle, is that when she was working with us in Iowa, one of the things that people have been doing is going in and in order to, from a drinking water perspective, right, in our local community, is going in and using activated carbon to avoid taste and odor, but which is not regulated, but consumer demand, right? So consumer demand, and then trying to remove the precursors for disinfection byproducts, which are regulated. But during the course of that, we were doing a study and we found 56 different pesticides that were also being removed during the process. And so these are not regulated, but they would have been in our drinking water. And so going in, and this is a place where the rules and regulations that are designed to remove one thing are actually having a cascade of benefits. And I hope this is something that we can think about from a green infrastructure perspective and emerging contaminants, is that we can go in and better connect the human health benefits associated with this, because this is all one water. And particularly as we think about stormwater as a resource, And that's going to be a place where it's recharging into our groundwater and it's impacting our drinking water sources and beyond just the ecology of our streams.
Well, I think there's a difference between detailed basic research and applied research that's more problem-driven. I think what's really important is that, excuse me, that we've identified degradation in receiving waters and natural water bodies from a biological community perspective, from an ecological standpoint, and then tried to find out what's maybe contributing, what anthropogenic factors are contributing to that degradation. It leads to an investigation of the chemical mixtures that some of these degraded communities are exposed to. And eventually, if we get lucky, we find a potential source, let's say through a discharge pathway or an effluent pathway, be it stormwater, point source discharge, agricultural runoff, urban runoff, et cetera, et cetera. I think that's where we begin to need to have a conversation about reasonable approaches to treating the problem once we've identified potential agents. And I think a lot of the applied work we've done on stormwater and CECs in stormwater is looking at existing opportunities, iron-enhanced sand filters, biochar, things that are used to remove a known pollutant or abatement of phosphorus, and see if there's an added benefit based on what's in the primary literature about affinity for organic compounds to these different biofiltration media. I mean, that's kind of what we've been doing, and funding sources often require to have that connection. So I think that role of applied research is really important, and it's helpful when practitioners and organizations that are involved in on-the-ground treatment are involved in sort of writing those RFPs, focusing on the applied research, bringing together researchers and the kind of basic and the applied questions they have about, you know, implementation at the same time. So I think it's really important for lack of a better term, different stakeholder communities to have an input into the nature of the funding mechanism that's going to be put out there for funding applied research.
Yeah, I can chime in from the kind of practitioner side of things. This is a really difficult topic for, you know, my taxpayers to understand and want to spend money on, essentially. And I think there is kind of a breakdown in communication from, you know, the academic to the practitioner, and like, how is this practical and useful? What can I do with this information? And one thing that has been really effective and striking to talking to, you know, just any general person is that our stormwater, the chemical makeup and all the pathogens in it, it looks more like wastewater than it does surface water. That's striking. But then we have this fundamental misunderstanding of, you know, what is stormwater? Everyone thinks stormwater is just treated at the wastewater treatment plant. So, you know, one thing we're doing right now, we're commissioning like some interactive displays showing the difference between like stormwater infrastructure and wastewater infrastructure, and tabling at public events. And, you know, here's where your wastewater goes, here's where your stormwater goes, and here's all the contaminants in them. So it's really simple tools like that that kind of start making a complex topic more digestible. And then how we have to, I guess, talk, you know, we're not focused on contaminants of emerging concern. Maybe that's 1% of my time in funding that I could dedicate to that. But I always trying to build it into everything we do. What I can do is I can design, you know, my structural BMPs in a way that facilitates applied research. We can pool our funding in places like the Minnesota Stormwater Research Council that supports these bigger efforts. So it's always brought up as, you know, something that's coming down the pipe, something we're looking at, we're just starting to invest in, you know, there's a lot more to learn. So kind of that's approach we've been taking at the Coon Creek Watershed District.
I can add just one thing to that, is that this is kind of brought up in the scaling of from a technology perspective too. It's like I mentioned, you know, cool, right? NSF, National Science Foundation, likes to fund stuff to come up with cool basic research this scale, right? And then practitioners go in and say like, well, have you tested it out in the field? No, we haven't, right? And there's kind of this dearth of how do we get from there? You know, that's the classic in business development is the innovation valley of death, right? How do we go from that type of thing? And so I do see opportunities and this like idea of pooling resources and bringing that in so that like multiple stakeholders do have skin in the game, and we see the benefits associated with that, so that you can take fundamental research that's really cool, but maybe not right to the scale of implementation yet, and bringing it to so it's kind of more shovel ready.
So I want to actually add, Justine kind of jumped ahead and answered a question, answered a question I was going to ask, but part of the question that builds onto that and the original question is, with whom do we need to connect this information? So we talked a little bit about, you know, what we've learned, and I certainly learned something here today, but for those that aren't in the room, then those that aren't going to see it online one way or another, with whom do we need to connect to get this information to them to help move it forward? Or who's missing from the table would be another way to phrase.
Well, I would add that it would be helpful if entities that are doing stormwater regulation at the national level were willing to expand their funding profiles to think about unregulated issues like CECs and stormwater, and making it available to researchers who are interested in doing applied research to try to solve the problems. Part of the problem with waiting for regulation before you try to start to solve a problem is that it goes unaddressed for a long period of time, which is kind of where we're at. The analytical methods that were developed in 98, 99, and the national surveys that were done by USGS scientists in the NRP program and elsewhere, and published in a broad survey of the occurrence of CECs nationwide, has been with us for over 2 decades. And we still are thinking about funding to try to understand this problem enough to try to abate it or ameliorate it in the environment. So that's one of the players that needs to be at the table.
I would say a kind of a low-hanging opportunity or a low-hanging fruit opportunity is potentially working with city engineers, because they're oftentimes the people that are getting like prioritizing stuff, right? And I've worked with a couple of communities in our area, and sometimes it's just not a priority. And sometimes you have a champion, and it is, you know, they want to just grab the bull by the horns. And so it's like that discrepancy between there, it's like sometimes it's just a level of awareness. And so sometimes if you have that champion, that's amazing. And sometimes the others represent an opportunity to learn a little bit more about it.
Who else do you think should be part of the conversation, or who else do we need to get information to?
Yeah, I mean, I think a big missing piece is maybe some of the higher level decision makers, our legislatures, and things like state agency folks that are involved in writing grant RFPs. Things like in Minnesota, you know, that might be the LCCMR and the Clean Water Fund Council, in making it easier to use some of this funding for applied research and monitoring when a lot of these activities are deemed ineligible. And maybe for certain pollutants, we're beyond that. Things like TSS and phosphorus, yes, don't spend money monitoring those. But for things like emerging contaminants, you know, we're at a different stage in learning about and addressing those pollutants. So I think there's maybe some room to expand some of those funding opportunities.
I think the only thing that I would add there is space there to have what I would call knowledge transfer, technology transfer professionals to help connect researchers and the things that we're learning in research and get it to practitioners and the public and legislators and decision makers in a format that they can understand. So I think there's space there to do that better. That was an in-person question. Let's take an online question.
What do you think are the differences in performance between biocells with an engineered soil mix versus a rain garden with an intact soil profile?
I guess it depends on what they mean about intact soil profile. I would assume that most, like if you're following the Minnesota Stormwater Manual or in most of these, right, there you have a mixture of sand, compost, and some residual native soil that's mixed in there. And those still work really well for a lot of things, right, that are good for removing pathogens and particle-associated things, and to a reasonable degree, stuff will still adsorb onto those, right? So it's not like it's terrible. Our idea here is to try to improve it. Removal, particularly for those more water-soluble, hydrophilic contaminants, right? Fancy way for saying water-soluble, but still maintain the hydraulic conductivity, right? And which is, it's reasonably difficult to succeed. And having a really high percentage of sand is not going to remove a lot of contaminants, and it also is not going to support as much biology as something elsewise, but you don't necessarily want to have just tons of compost that are going to leach out.
Other thoughts?
I do know I will expand and maybe emphasize something Greg said. I do think it depends because soil is highly variable. Even engineered design systems within the US, if you look at stormwater manuals, even in Minnesota, but also across the nation, there's a lot of variability in what is engineered media and what qualifies. And of course, in many places, we're starting to incorporate things like biochar and other amendments that are even beyond the scope of what's currently engineered. So that's a tough question to answer because it does highly depend. That was online. Who has an in-person question? Oh, we've got one back here.
Thank you. So you mentioned biochar a couple times, and I'm curious if you could talk about the relationship between biochar and sort beads, and if those are kind of similar strategies and if they can work together. And then also, we have the biochar facility being constructed in the city now. So do you see that as an opportunity to kind of further your research there, or is it kind of similar but separate?
You talked about the beads.
Sure.
Yeah. I think if we have a local source of biochar, there is the opportunity to incorporate it in stormwater practices and have some more field testing opportunities with something that's sourced and consistent across various areas, and because it's local, it'll make it easier to do so. I think there's more and more evidence that biochar, if nothing else, is a really good sorbent, right? And that doesn't solve all the problems. It's going to have certain capacities, but if it does act like the absorption beads at all and retain things enough that they can then be leached out and biodegraded slowly, that would be great too. That's just going to take some study to figure out if that happens, but you can do a comparison, Greg.
It would be cool. I know, actually, Bridget Ulrich at University of Minnesota Duluth is at the Natural Resources Research Institute there and has been doing a lot of stuff with biochar and stuff, and it's really cool because it's a resource development opportunity too. And in Minneapolis, they will use the wood waste for making cutting boards and potentially use it for biochar and things too. I mean, we use powdered activated carbon just because it's commercially available. You could easily powder biochar. One of the things that we've done in another related project is that black carbon is kind of a funny thing, and most of the stuff that is in a Brita filter that's activated carbon and stuff that would be in a commercial drinking water filters is very sorptive. And sometimes if you want to combine that with biodegradation, sometimes it's actually better to have a biochar that doesn't actually work as well as a sorbent because it means that it can be more bioavailable to the biofilm that's on it. So there's some of the imperfections that make activated carbon better in drinking water treatment actually make it better for a passive stormwater system for the biofilms that are growing on it and how they're kind of providing in situ bioremediation. So it's particularly if you're talking about putting this at scale, it is a way more sustainable source, because a lot of the stuff that is in drinking water comes from like coconut shells and stuff like that, which is great for that type of need, but not for if we want millions of pounds of it.
Yeah, so I can chime in. At the Coon Creek Watershed District, we have been using biochar in our filtration practices since 2020. So we have about 5 years of performance monitoring data, and we have, very large regional filtration systems. We did source our biochar commercially, so it had to get trucked in from Michigan or elsewhere. So it's expensive and not the most sustainable. So we're really excited to have locally produced biochar available soon. But we started integrating this into our sand and our iron sand filters to try to optimize these filters for bacteria and pathogen capture, because we are impaired for E. coli and we're regulated for it. But with the hope that we can hopefully, capture CECs and heavy metals and other things. So we designed our filters, a couple of them with kind of head-to-head multi-cell, so we can do some applied research and some comparisons. And we partnered with Sarah Elliott's group at USGS and Andy Erickson's group at SAFL with some LCCMR funding to start measuring a suite of hundreds of contaminants of emerging concern and doing some paired influent and effluent testing. We presented some of this work up at the Minnesota Watersheds Conference just a couple weeks ago, so the presentation will be live up there soon, in a couple months probably. But I guess spoiler alert, it's really hard to have statistically valid significant differences when you're talking about 6 or 8 paired stormwater samples and the huge range and the small amounts of these contaminants detected. But you can see the patterns in all the summary graphs, in that there's more contaminants and higher amounts of them cumulatively in the influent, and they're being reduced even through our regular sand filters or sand iron filters, and you add biochar and the amount detected and the numbers are less. But when you look at any one individual and you're not sampling the exact same parcel of water, influent, effluent, some of those statistical kind of power breaks down. But we're really encouraged by the preliminary results. And then one thing that we just did is we're taking media samples and looking at the microbiological communities. So those results aren't available yet, but stay tuned. Sarah Elliott is here if you want to talk to her more about it. She's the expert on that.
Yeah. And I would add that, you're going to get biofilm colonization of biochar that's in the field and stuff. Kind of our goal with these things is to bring your microbes into direct proximity, because sometimes you'll have contaminant over here and microbes over here, bring them together more so, and then trying to get a lot of those properties in the same thing, as well as being able to jumpstart with the exact microbes that you want right away. So yeah, some person could, if there were enough of them, add them as a layer, or you could go in and put it as a post-construction amendment or something like that. I mean, it's still kind of very vision-y at this point, and biochar in and of itself works really well. And if you wanted to combine that with these, so that's why it's really cool to hear about the.
Yeah, I think that connection between identifying what might be colonizing the biochar media and facilitating biological transformation can be tied to your approach with beads. I think there's maybe an analogous system would be soils that are stockpiled for a long time on a site and then redistributed for landscaping. If there's no active mycorrhizae community in them, it takes much longer time for that growth dynamics to approach a desired endpoint. And I think the opportunity here for seeding new structures that are put in is, a real potential. It's one of my takeaways from today's seminar, really.
Yeah, and I did want to chime in too, this idea of plants. So plants are kind of annoyance when you have regional sand filtration systems. They're a maintenance headache. When you need to till this media, make sure your iron isn't crusting. You don't want those tap roots interfering with your drain tiles and that sort of thing. But I will say, anecdotally, we can grow a lot of plants on a biochar amended iron sand filter bed. Generally, some people struggle with growing plants when it's just sand and iron, and we don't want to add compost and have that leaching. We put the biochar in, and the plants will come by year 2, 90% coverage. So whether that is a maintenance headache or a really good thing for biological processes and supporting mycorrhizal fungi and all that, to be determined, but I would be really interested in kind of learning more about how our plants are impacting performance.
Let's take another online question, please.
Do you think it would work to place degrading wood particles into our bioretention systems to allow the wood-based fungi to degrade organic chemicals?
Yes. So, I mean, one of the reasons why we added or put in the wood flour into here is the wood dust can act as a maintenance substrate for the fungi to stay in there for a really long time, right? You could kind of achieve some of that by going in there and adding wood chips or something. Then you'd kind of, that's what like a denitrifying wood chip bioreactor is for agricultural drainage and such. Obviously, you would have to go in and keep it saturated if you're trying to do nitrate removal and stuff. If you're trying to grow fungi, they want to maintain aerobic conditions. So depending on where you are and everything, understanding basically whether it's air or it's aerobic or anoxic is really important. So there's lots of different ways you can kind of skin this cat, right? In terms of putting the things together. What we're trying to do here is make it so that hopefully it can be easier and faster. But if you have things that are like really big wood chips, one of the things that can happen is that when they do break down, they really compact. It's like being on a playground and you see how much the stuff is, and there are people always supplementing wood chips on a playground.
Other thoughts? All right, let's take in-person.
Floodplains, so specifically marinas. We have a boatyard, which like contaminants are tremendous because we do so much work on the boats. So the idea I'm asking about is like, as we're doing strategic planning for the marinas and for taking over additional marinas, we're right on the river. It obviously floods. The boatyard floods. So we need a speedy thing to try to remediate, and then also a design that's going to push the water to be remediated away from the river. And all they keep putting in now is because the trees just fall into the river. A lot of stone riprap there. Maybe we could layer beneath the stone riprap. Can you give me some ideas about what's the best way to work with a marina and a floodplain?
I was going to say that's interesting hydrology and different than what you probably normally think of with stormwater and runoff. And that's, you've got runoff coming from the land through the marina and then back the other way. I'm not a hydrologist, but that's a really interesting problem. And if you can find a consultant who's got a good design plan there, they can make some money, that's for sure.
I know I would point to Ryan Winston at Ohio State University actually has done some research. He published a paper, maybe even 2, on some work around stormwater runoff related to marinas. And I think coincidentally, he did his work in Duluth, or Superior, Wisconsin. And he didn't call me when he was in town, but Ryan Winston, yep.
He was on the slide at the beginning.
Yeah, he was in one of the slides. I can give you contact info if you're interested. But he did some work related to stormwater and marinas and marina contaminants and chemicals specific in that type of runoff. So there's some, I can lead you to there. Let's take another online question.
Early in his presentation, Greg mentioned something about accumulation of CECs in the landscape. Is this similar to phosphorus pooling in ponds, wetlands, and basins? How much of a concern is this for CECs?
Yeah, so I kind of tried to set that up from, the two contrasting things that we don't want to have happen in going in and like if we're directing polluted water into a bioretention cell or any type of green infrastructure system or a pond or anything like that, right? You don't want it to pass through because that means it's not working, but you also don't want it to just accumulate, right? And to go in and to be in there, which is one of the reasons why we're interested in biological processes or potentially other abiotic processes, but like why we're interested in breakdown processes in situ so it can sustain removal. And so there are some things that for which you potentially, I mean, if you don't have, or for something that wouldn't be broken down, I mean, the classic case that always is to me most scary are things like metals, right? They're not going to break down. To a limited extent, some phases might be taken up into plants or something, but again, that's just going to be recycled, right? It's not going to, there's no opportunity for breakdown there. And so that is an issue, right? And for some types of organic contaminants, you wouldn't want, you wouldn't want accumulation, but you also don't want pass-through. So somewhere in the middle, you want to capture and you want to degrade to sustain.
And I think some of our infrastructure is designed to capture. So like Greg mentioned ponds, right? And we know, for example, pHs are in the bottom of stormwater ponds. And my guess is there's a lot of tired dust in the bottom of stormwater ponds too, and that means there's a lot of the tire-derived chemicals down there. And I think we've only started to scratch the surface as to what's being degraded and what's being deposited, and is the system anaerobic or aerobic? That's all going to affect the persistence of these chemicals. And so, yeah, until there's some regulation that makes us look for them in detail and understand what's going on, it's going to be an academic exercise for the moment. But
Yeah, I'll chime in. I'm worried about this idea of capture and accumulation, and I was kind of really interested in Greg's talk about separating the timescales of treatment, that we have this kind of the first flush, make sure it infiltrates, and then use maybe the antecedent, the dry periods where the microbiology can break things down and transform things. But I'm worried, like, I've built these giant inline media filters with biochar. What is in there? And maybe they're working really great at removing first flush, so I don't get these toxic plumes of things like 6-ppdq or something like that. But what about kind of the chronic and sublethal impacts on biota downstream? Am I just slowly releasing really small amounts of some other chemical that could be a problem? So I think it's something that needs a lot more work. And and like what kind of Randy asked, what about the plants? Should I be harvesting and baling and landfilling those? And is that a way I can maybe get rid of some of these things and some heavy metals, or we just let them decompose and that goes right back into the media and then they grow up again? So perhaps there's some other areas where we can kind of intervene with these, relatively simple, cost-effective management actions.
It almost gets a little philosophical because you're like, what is the environment, right? We built these pieces of pollutant removal infrastructure, but if you make a pond, these things get integrated into communities and things like that. And if there's people fishing in them and there are people like, this is now, it's part of the environment. It's not just a pollution treatment control device. And so it's like, removal is relative.
And I think that brings up the role of stormwater treatment ponds and infrastructure in urban and exurban wildlife biota. Major habitat source for amphibians, reptiles, and mammals, vertebrates. And I guess the real question is, are we sequestering the compounds, like PAHs and metals maybe in the bottom sediments that can be removed at great cost and treated and gotten rid of? Or are we just enhancing transformational dynamics like Greg suggested, where the compound is immobilized in a plant, but then released at some point, transformed by bacteria, and the original compound or some congener of it continues to basically proceed through food webs, through the environment? That's something we really need to understand, especially when it comes to these trace organic contaminants. So, sorry, It's complex, and we need more information. And that's another takeaway from today's talk, I think.
As much as we do need more information, we do like to understand, and that discovery is really important. I mean, one of the things should be, as Bill talked about, an endpoint, right? If we can demonstrate toxicity or something, and that may be our end goal for a lot of things. Like, if you go to transfer to another thing is like harmful algal blooms, right? You're not necessarily looking for the bajillion different potential algal toxins. You're looking whether it's causing a toxic effect with a ELISA binding assay, right? Is it causing a toxic effect? So if there were something magic with, I think that's a cool next frontier type of thing. It's like, is there a whole, magic probe toxicity thing that we could go in and like look for in terms of organic contaminants or something in that area? There are things like effect-directed analyses to try to bridge some of these things and whatnot. And one of the, trying to merge together the big data revolutions of transcriptomics and things like that from a biology standpoint, and these high-res mass spec cheminformatic metabolomics and stuff, putting them together, I think is a really powerful research frontier area, but it should be hopefully moving towards the so what question, right? How is this impacting our waterways?
Greg, thanks for taking us down the mechanisms and not just the chemistry, but the biology. I really liked, Richard's add-on where it's, we're looking for the degradation processes and not the transformation processes. It got me thinking about, this is a competition for space and time in the future things that we're going to build. And whether it's a plant or a fungus or a bacteria, we're looking to curate a group of, life forms that can do that degradation. So it got me thinking more about the strategy for scaling. So your tech development, for these particles, I thought was great, and to think through the chain of events that you would need to do to design or curate that mix that we want that can do all the things. Could we kind of talk to me about more of the strategy on the scientific side to get a lot more of those potential candidates out and competing together? I was just thinking, as Bill and Justine talked about the low concentrations and the ubiquitous nature, if we gave you a list of 20 really toxic, gross things that we want, how do you spin up the science to give me the 100 different bacteria and fungus that are going to do the work to degrade?
Yeah. We'd like to sometimes refer to the hypothetical dimethyl death. Right, we don't know what it is or something like that. I guess one of the things is that, we effectively do wastewater treatment, and we don't necessarily go in and have just one optimized bug, right? That's in it. And in fact, the way that nature in ecosystems builds resilience is through diversity, right? We can probably learn something about that for our own society too, but that's how we build resilient systems. And so we're essentially curating a little ecosystem here. And because there are such complex mixtures coming into here, as there are in wastewater, right? In a biological treatment process, there's this thing called the minimum substrate theory, right? You have to have a certain amount, otherwise you're not going to upregulate the enzymes in order to be able to degrade it and stuff. But with the complex community that evolves, you can go in and make it so that there's a lot of things that could potentially be biodegraders. And really kind of the fundamental crux of what we're trying to do is very much like wastewater treatment, is to make it so that the chemicals spend more time in the system than the water does, so that there is a greater opportunity to experience chemical degradation via these different biological processes. So did that answer your question? Okay.
Well, and I think another thing we need to think about too is a lot of times with the chemicals, we find there's a problem and we're trying to develop technology to solve the presence of the chemical, we also need to think about the upstream and say, okay, do we need this chemical? Are we using it properly? Should we have a substitution? You know, I have no idea what's going to happen in the Pacific Northwest, but one solution would be, could you make tires without 6 ppdq? I have no idea if that's possible, right? But that would solve that problem, because then you wouldn't have the salmon, if you're not putting the chemical in, if you turn off the tap, and this has been argued for PFAS and for other things as well, is we need to maybe rethink some of our, not infrastructure, but our commercial products.
One of the challenges with the analogy for wastewater treatment is residence time that you have in the treatment and what end result you're going to look for. Trace organics, for example, can be removed more effectively in a lot of wastewater treatment plants if they're running at a lower percentage of their design capacity, because it has to do with residence time. And of course, that means what, in many cases, what you're looking at ends up in the sludge or is degraded or transformed and ends up in the sludge, so it's just in another place. But that's also going to be a challenge for stormwater systems, because we talked about fast infiltration, movement into, we're talking about groundwater, but what about the residence time necessary to treat it or to abate the compounds or transform them? I don't have an answer for that, but I think that's a really big question too.
Right.
I don't know, Greg, what do you think?
I mean, because, yeah,
It's
that brings up a
huge.
big point that, I mean, wastewater treatment plants do not effectively remove a lot of emerging contaminants.
Right, because we just working with the city of Hutchinson, Sarah Elliott and I and Heiko Schoenfuss looked at membrane reactors as opposed to oxidation ditch dynamics and looked at the microbiome and the results. And that's very specific technology that's been put together for wastewater treatment. I don't know that we're close to that yet for stormwater, but that's sort of what we're talking about as a group here, about biochar, iron enhanced sand filters, rain gardens. I know compost is, a bad word in some stormwater circles, but it is a really nice biologically active medium. And so maybe what we're going to find out is something like compost is actually maybe better for initial capture and degradation of trace organics, whereas it's not necessarily great for nutrient abatement or phosphorus abatement. So I don't know.
I'll say
That's just speculation on my part.
Andy and I are running an experiment downstairs that shows that's not the case.
Okay. One of the fundamental challenges of these is that you're trying to optimize for so many different things, and you can't do it all.
Yeah, I don't know if the entire audience has an appreciation for just how many compounds we're talking about. But we analyzed for, it's been a while now, since April, since I've been involved, but we'll look at 400 to 500 compounds. We'll find 30 to 50. Sometimes as few as 10, sometimes as many as 90. They're not always the same 90. It's that's a real challenge. So I just want to make sure everybody understands what we're talking about.
Any other thoughts on that question? All right, well, we are rapidly running out of time. We're going to go to our closing question. I'm going to ask each of our panelists to give their key takeaways today, either re-emphasizing something you want to highlight as a key takeaway or possibly something we didn't get a chance to cover that you want to make sure we get in before we close. I'm going to start again on this side, just as we did introductions. Bill, what's your key takeaway? I know.
Yeah. I think two things. One is the problem is complicated just by sheer number of chemicals, the fact that there isn't regulation that drives things. And I think a really key point here is that communication is really important too, from public to practitioner to taxpayer to facility manager to regulator to academic. There's lots of spaces where you can lose communication, and if communication is better along that pipeline, we'll get better solutions.
Wonderful, Bill. Justine, what are your key takeaways?
Yeah, I guess I'm going to go back to one of the last slides that Greg showed about the hierarchy of treatment complexity and kind of we are here in the bottom corner, stage 1 of treating conventional pollutants, and that we need to move up that and, getting to stage 3 where you start playing with these, media amendments and things like that and deliberately targeting pollutants in known hotspots. I think it's going to take all of us to get there, and we can collectively kind of pool our resources, and you don't have to be an expert to participate in that applied research, and we can all learn something from each other, so.
Well said, Justine. Richard, what are your key takeaways?
Well, I'd like to build on that last point Justine made, because I also focused on that slide, and the risk component on the x-axis was something that I thought was really interesting. And maybe we need to start thinking about the numerous compounds we're finding and the frequency of those compounds and the associated risks, the things we really need to focus on, and where we need to up our treatment game, if you will, which was sort of that y-axis on that graph. So I think that's really important. The other takeaway was the focus on mechanism, which has kind of been a mantra. I like the point you made about the 3 M's. A lot of my career's been stuck on the mechanism part until I started doing more applied work. And I do think it's important, though, if we're going to have effective treatment technologies, that they're based on a sound mechanism. We don't need to understand, perhaps, the entirety of the ins and outs, because you talked about wastewater treatment. But we do have to have an understanding of whether it's maybe bacterial-based or fungal-based or a combination of that, a community-based degradation of these compounds that will help us with building new infrastructure in the future. So.
Very nice, Richard. And before I give Greg the last word, I want to reemphasize a couple things. One thing that Bill said, something Justine said, source reduction. So we talked a lot about treatment and removal and even degradation, but source reduction, I think, is a key part of this puzzle. And also thinking about, is your jurisdiction the source of potentially some of these chemicals for downstream partners? So think about that. But Greg, you get the final word.
All right. I was actually just going to play out source reduction, because that is typically the cheapest, easiest thing that we can do from a mass load in our watershed. That's so important, particularly for so many of these emerging contaminants. It's also really quite challenging because, I mean, you don't really think of necessarily like a hardware store as being a dealer for pesticides, right? Like, but collectively, what goes on in people's everyday lives are what's impacting stormwater. I think it is very important to understand mechanisms so that you can kind of unlock the black box and what's going on inside bioretention, because with that, you can then try to situationally optimize in different directions and make improvements. But at the same time, we shouldn't, wait for action, right? We know how to do a lot of really good things. And so there's a lot of important opportunities for scaling and improving our systems. And the reason why we put that kind of framework was because we want to try and improve it, but do it in the way that makes it so that we can improve the most water with the amount of resources that we have available.
Very well said. Thank you so much, Greg. And that will conclude this month's Minnesota Stormwater Seminar Series. I want to thank you all for joining us today, both in person and online. I encourage you to check out our website and our schedule. The speaker list for 2026 is super exciting. We're going to have some very fun topics and some very good discussions. I encourage you to check out a future Stormwater Seminar Series. Also go to our YouTube channel. All of these are recorded and online and available for free, so tune in there as well. Thank you so much for joining us. I hope to see you at a future one. Thank you.